Good afternoon, everybody. My name is Tommy Soares. I'm the Assistant Director of the Purdue Institute of Inflammation, Immunology, and Infectious Disease. And we welcome you today for the Purdue Lecture Hall series. We're joined by none other than Molly Youse from the Department of Medicinal Chemistry and Molecular Pharmacology in the laboratory of Dan Flaherty. Molly is doing drug discovery for Neisseria gonorrhoeae, and I can't wait to hear this story. Molly, thank you so much for doing this, and welcome to the Purdue Lecture Hall series. Welcome,
All
Molly.
right, thank you. All right, so thank you, Tommy, for the introduction. As he mentioned, I'm going to be sharing a little bit about the research that I do in the Flaherty lab at Purdue University. And to sum up what I do in my research in one sentence, I'm trying to make drugs that are effective against the bacteria Neisseria gonorrhoeae. So first, I just want to give some background on how I got to Purdue. And it actually started way back in high school when I thought I wanted to be a pharmacist. I was really interested in medicine and helping people. So in high school, I was able to shadow some pharmacists in a couple different local pharmacies in clinical settings, things like being in a hospital, and then also in like a CVS pharmacy as well. And when it came time for me to apply to colleges, I decided to apply to some colleges that had a pharmacy program and some that did not as well, just to keep my options open and just so I could kind of figure out what I wanted to do after I graduated. So I ended up going to Vassar College, which did not have a pharmacy program, but I decided that after my 4 years at Vassar, if I still wanted to be a pharmacist, I could go to pharmacy school after getting my bachelor's degree. So while I was at Vassar, I majored in chemistry, and I spent one summer working at a pharmacy and then another summer working at a research lab at Vassar, conducting chemistry research. And that summer, I realized how much I enjoyed being in the lab. As much as I thought it was really interesting and exciting to be behind a counter filling prescriptions and knowing what medicines are needed for different types of patients, I thought it was even more exciting to be kind of a step even further back and be the ones in the lab actually doing the experiments to eventually get to the medicine. So because of that, I decided to focus on medicinal chemistry after graduating from Vassar. And that's what led me to Purdue. So I applied to the MCMP department, the Medicinal Chemistry and Molecular Pharmacology department at Purdue, because it has a lot of different research interests, a lot of different types of things that you can do within the department. Include doing research on viruses, working with bacteria. Some people work with DNA. Some people do your typical chemistry. That is mostly what I do, but there's also some computational work that you can do, modeling proteins and drugs in computer software. Some people work with animal models and live mice. So all of these things interested me. I wasn't quite sure where I wanted to end up within medicinal chemistry, but I knew Purdue would be the right place for me to explore all these options. So I want to explain a little bit about what drug discovery really means, and for the entire drug discovery process, what part my research is focused in. So there are 4 parts of drug discovery, starting with target identification, and that means what are you developing the drug to be used for? So in my case, I want my drugs to interact with a protein called the carbonic anhydrase in the bacteria Neisseria gonorrhoeae. So that target was already identified before I entered the lab. So the professor, Dan Flaherty, who runs my lab, he had already identified this protein as a target of interest to develop drugs for. Something else when you are identifying a target is, do we want to activate the target or do we want to inhibit the target? So if we want to activate a target, that means we want the protein to be doing its job. So sometimes it may need a little help. Or do we want to inhibit it? Do we want to stop the protein from doing its job? So in my case, for my research, I want to inhibit the protein. If I can inhibit the protein, then the bacteria is not able to continue to spread its infection throughout the body. Specifically, what I'm doing in the lab is I synthesize molecules. So like I said, I'm doing a lot of chemistry, setting up chemical reactions that you may learn about in chemistry class. And then I also test the molecules in vitro, which means I'm not testing them on humans or on animals, but I'm testing them using test tubes, petri dishes, things like that. And then once I test these molecules that I have made, we evaluate their efficacy. So we see how good of a drug they are. And then from there, we can go back to the drawing board and we can synthesize even more molecules based on the discoveries that we found through testing them. So that's my part in the drug discovery process. Eventually down the road, hopefully after making a lot of molecules and testing them, we get a molecule that's good enough to then be further developed and then eventually going into clinical trials, things like that, before eventually making it to the market. So the reason that I'm targeting a protein in the bacteria Neisseria gonorrhoeae is because gonorrhea is becoming a really big problem, not just in the United States, but worldwide. So there are several complications that can arise if you get the infection and it goes untreated. So some of these things include pelvic inflammatory disease. The infection can spread throughout the body, through the bloodstream. There can be a lot of pain associated with it. And in 2019, there were 616,000 new cases that were reported just in the United States. However, there's probably even more cases that go unreported because a lot of people actually are asymptomatic. So we hypothesize that it's actually a lot more prevalent than we even know. And worldwide, the incidence rate is about 21 per every 1,000 people. Now, the problem with this is Neisseria gonorrhoeae is becoming more and more drug resistant over time. So on the right here, you can see the timeline of different molecules, different drugs that no longer work against Neisseria gonorrhoeae because the bacteria has developed some sort of resistance for it. So you can see way back in the 1980s, penicillin and tetracycline no longer worked against the bacteria. In 2007, ciprofloxacin was no longer recommended. In 2012, cefixime was no longer recommended. And at this point in time, as of 2020, the CDC now recommends there's only one drug that is effective against the infection. So this is a problem because history repeats itself, and if we don't develop new drugs soon, it's a very real possibility that there won't be any drugs left to treat the disease. So my two main research questions I'm going to be addressing today is how can we make drugs that can be used to treat gonorrhea? And then how can we test these drugs to see how effective they actually are? So as I mentioned, my target, I'll be referring to it as NgCA, and carbonic anhydrases are proteins that are found in eukaryotes, algae, and bacteria. So we have carbonic anhydrases in us as well. They're called HCAs, or human carbonic anhydrases. And these proteins are important for physiological processes like maintaining the pH and the amount of carbon dioxide in our cells. So as you can see down here, the reaction between carbon dioxide and water through the help of the carbonic anhydrase becomes bicarbonate and this H proton. So the way this reaction happens is by using this zinc ion. So here is a picture of what the NgCA protein looks like. And you can see this gray dot in the middle of it is a zinc ion. So what happens in the catalysis of this reaction is water interacts with this zinc ion. Dioxide comes along and interacts with the zinc and the water as well. And then we're left with the bicarbonate and this H proton. So because this all is facilitated by that zinc ion, if we block the zinc ion from being accessed, this reaction can no longer proceed. So that is essentially what I'm trying to do with the drugs that I make. I want to block that zinc ion. And the way that I'm going about doing that is drugs that are already approved for human carbonic anhydrases, and I want to somehow change their structures so that they are effective against the bacteria. So here I have ethoxyzolamide or EZM on the left and acetazolamide or AZM on the right. So these drugs are currently used to treat epilepsy and glaucoma, among other symptoms and diseases as well. If you zoom in close, you can see that EZM here bound or interacting to the human carbonic anhydrase. You can see that right here is the part of the molecule that interacts with the zinc ion. Similarly, acetazolamide, again, this part of the molecule is what interacts with the zinc ion. So that part of the molecule is this SO2NH2, which is a sulfonamide. So these sulfonamides, by interacting with the zinc ion, is how these drugs are effective against the protein. So what I want to do is take the structures of these FDA-approved drugs and, like I said, somehow modify them so that they are effective against the bacterial protein. So the way that I will do this is I'll test the molecules, I'll synthesize molecules, and then I'll test them and evaluate them. So like I said, the most important part of the molecule is the sulfonamides here. So what I will do instead is I modify the structures coming off of this part of each molecule. So if you've taken a biology course or a biochemistry course, you might be familiar with this lock and key model of an enzyme and a substrate. So in general, for an enzyme or a protein, it has a binding pocket here that a substrate or a molecule can then fit into. So to illustrate this a little bit better, if you look at this picture of the human carbonic anhydrase, you can see that in its binding pocket here, the acetazolamide molecule fits really well. For NgCA, the bacterial protein, its binding pocket fit is a little bit different. It's shaped slightly differently so that it can bind other molecules to it. So in this case, you can see that this molecule looks very similar to acetazolamide. The only difference is it has this big cyclohexyl group to it. So if we try to fit that same molecule into the binding site of the human protein, you can see it doesn't really fit well. So because of that, we can say that this molecule has selectivity. It is selective for the bacterial enzyme. It fits nicely. It can bind to the target very well. However, it does not bind so well to the human enzyme. So this is ultimately what we're going for. We want to develop a drug that is selective for the bacterial protein that does not interact with the human protein. If we have molecules that bind to the human protein, that is what leads to side effects. So next I'm going to explain two of the ways that we test our molecules. So we test them against the protein NgCA itself, and then also against the bacteria as a whole. Because if our molecules aren't effective against the bacteria, then they won't be able to do their job if you have a bacterial infection in a human. So I'll explain the two different tests to do that. As I mentioned, we know that the carbonic anhydrase helps the reaction to form HCO3 and H+. So what we can do is we can have a solution with carbon dioxide in it, and we put our molecules, our drugs, into this solution. We incubate it, and then we can measure how much HCO3 is actually produced. And by doing that, we know how well the molecule binds and inhibits the protein. So if a lot of HCO3 is produced, that means that the protein can still do its job, which isn't great for us. If just a small amount of HCO3 is produced, we know that our molecule is inhibiting the protein, so the protein can no longer work, which is what we want. The other thing we do is when we test the molecules versus gonorrhea, So we can grow gonorrhea on a petri dish like this, and then we can dose the bacteria with our molecules at different concentrations. Then we can see what is the minimum concentration that stops this bacteria from growing. So by doing that, we can see how much drug it will actually take to stop the growth of gonorrhea. So to explain this a little bit more, when we are testing our molecules versus just the protein, we are measuring this using what is called a Ki, or an inhibition constant. So this is the concentration required to inhibit the protein by 50%. And we want this number to be as low as possible, because the lower amount of drug that is necessary means it's better. In a real world setting, you don't want someone have to take grams and grams of ibuprofen just to stop a headache. So the same type of thing here. We want as little drug as possible. Molecule 21 and 29 both have really low KIs at 4.1 and 8.4. Also, you can see that their KIs are higher against the human protein, which again is what we want, because we want our molecules to be effective against NgCA and not HCA. However, on the opposite hand, you can see that 40 and 48 are not very good drug molecules for us. They have high KIs against the bacterial protein, and they have low KIs against the human protein. So this is the type of molecule that we don't want to use. Similarly, for the MIC, the minimum inhibitory concentration, we want this number to be as low as possible as well. So as you can see, 22, 29, and 19 all have low MICs against the bacteria, ranging from 2 to 0.5. However, 58 and 59 are not so effective against stopping the growth of the bacteria. So again, 58 and 59 aren't great molecules for us to be using. So just some other examples. Some of our molecules are effective against the protein, but not effective against the bacteria. For example, number 10, you can see it has a pretty low Ki against NgCA, but it has a high MIC. So even though it is really good against the protein, it's not so good against the actual bacteria. However, compound 82, which just differs from compound 10 by an extra carbon here, this molecule is effective both against the protein and against the bacteria. Similarly, you can see that 68, it is effective against the protein, but not effective against the bacteria, with a high MIC of 64. However, 29 is pretty effective against both the protein and the bacteria. So while we were going through some of this data, we've made about 100 different molecules that we have tested. And we noticed that some of our molecules are really effective against the protein just outside the cell. So just the protein in a test tube with the molecule itself, it works great. However, once that molecule is tested against the bacteria, it doesn't do so well. So we're trying to figure out why that may be. And a hypothesis we have is that those drugs that aren't doing very well against the bacteria aren't able to actually penetrate into the cell. So if the molecule can't get into the bacteria, it can no longer be effective. So to test this hypothesis, we have tested how much drug can actually get inside the bacterial gonorrhea cell. So just to give a little background, there are known properties for drugs to effectively get inside the cell of E. coli. So for example, if a drug has this NH3, if the drug is flat instead of being having a globularity or three-dimensionality, if it's a pretty rigid molecule, and if it's amphiphilic, then it can easily get inside E. coli. So based off of that, we have our own hypothesis that there are also some properties or rules that a drug must have in order to get inside Neisseria gonorrhoeae. So the way that drugs actually get into bacteria is through something called a porin, which is kind of like a tunnel. So if these molecules are unable to pass through the tunnel, then they can no longer get inside the cell. So as you can see here, our protein, NgCA, is inside the cell, and we think that some of our molecules are ineffective against the bacteria because they're unable to travel through the outer membrane and the inner membrane to eventually reach their target in the bacteria. So the way that we test this, we have developed an experiment to see how much drug actually gets inside. So the way we do this is we grow the bacteria and then we add in our drug, and we let that sit for about 2 hours to give the drug some time to actually get inside. After we do that, we centrifuge it, which means we just spin around the test tubes very quickly in order to separate the actual bacteria and then any drug that did not get inside the bacteria. So we take any leftover drug out, and then we're just left with the bacteria and any drug inside the bacteria. Next, we break apart the bacterial cells, and then we centrifuge it again. So by spinning around the broken up bacterial cells, we can then separate any drug that was inside the cells from the bacteria. We can then put this on an instrument called an LC-MS, and this instrument allows us to measure and quantify how much drug was actually inside the bacteria. So we did this with acetazolamide and ethoxylamide. So acetazolamide is in green here, and ethoxylamide is in blue. And these bars show how much drug was actually inside the bacteria itself. So you can see that after 2 hours, and actually even after just 10 minutes, there is much more ethoxylamide inside the bacteria than there is acetazolamide. Ethoxylamide accumulates more in Neisseria gonorrhoeae and is more effective against the bacteria. You can see it has an MIC of 0.25 compared to acetazolamide's MIC of 2. So this kind of helped confirm our hypothesis. Maybe EZM is so effective against the bacteria and has such a low MIC because it's a lot easier for EZM to actually enter the cell. So our next steps for this is to test all of our molecules, and from there we hope to be able to establish some sort of properties or rules that a molecule must have in order to highly accumulate in Neisseria gonorrhoeae. And if we can do that, then we can better design more molecules to make if we know ahead of time what will make them successful. So another experiment that I've been working on is figuring out how quickly do the drugs bind to NgCA, and then how long it takes for them to then dissociate or unbind from NgCA. So to do this, I run an experiment called surface plasmon resonance, or SPR, to test binding. And this is important because we want our molecules to bind quickly to our target and stay there for a long time. The longer that the drug is able to be bound to the protein, the more effective it is. So to do this, we use an instrument that looks like this, and it has a little plate or a little chip here. And on this chip, we put our target, our NgCA, onto it. Then we put this chip inside the SPR instrument. And once inside on this little chip here, you can see we have the protein bound to the chip, and then we inject our molecule, and we can measure how quickly the molecule is able to bind to NgCA. And we measure that using a parameter called the K-on rate, how quickly the molecule binds. Eventually, the molecules will then dissociate, and that is measured by a parameter called the K-off rate. So by using the K-on rate and the K-off rate, we can see how quickly the molecule binds, how long it's actually staying bound to our protein before then dissociating. After running our molecule through this instrument, we get an output that looks like this, which is just different curves at different concentrations of our drug. And this, through some math that the computer software is able to do. Thankfully, I don't have to do it, but using these curves, the software is able to calculate a KD. So a KD then is a measure of the binding or the affinity of a molecule to our protein. And once again, the lower the KD, the better the binding of the molecule is. So in conclusion, I make molecules in the lab that are effective against the protein NgCA and are also effective against the bacteria Neisseria gonorrhoeae. We also have some molecules that are selective for the bacterial enzyme over the human enzyme. I also use an accumulation assay to determine how much molecule actually gets inside the bacteria in order to do its job. Then I use SPR to analyze how quickly the molecule can associate and dissociate from the protein. So that's all I have for you today. I hope you learned something new. I hope you found my research interesting. If you have any questions about my research or the drug discovery process or college and grad school in general, I would be happy to take any questions. This is my email, so feel free to reach out to me if you want to talk about anything that I mentioned here today. Thanks.
Thank you, Molly. This was fantastic. So exciting to see you do such great work, very sophisticated work too. I just want to say that it's a lot of experiments, it's a lot of comparisons and contrasts. I have a few questions. I hope you don't
Sure.
mind. Yeah.
Mhm.
And because I know that the audience primarily of this lecture is going to be high school students that are thinking about college. They're thinking about, what am I going to do? What am I going to study? I wonder if you could take us back a little bit to that time, and you can think back of when you were making those decisions and thinking about it. What was it that made you think for science and, again, this whole piece of pharmacy, and you wanted to shadow pharmacists? And what was it that inspired you to think, oh, maybe pharmacy could be cool? Or, can you take us back a little bit?
Sure. Yeah, so I always really enjoyed my science classes. In high school, I enjoyed chemistry and biology, but I also really wanted to help people in some way. But I knew that I didn't really want to go the doctor route. I knew that, I wanted to still be able to use my hands somehow, maybe. But yeah, I really wasn't sure. So if I could give any advice, I would say if you have an idea about what you want to do, maybe, or just any sort of inspiration, try to reach out to people who have experience doing that thing and see if you can shadow someone, or see if you can just email back and forth with someone who can give you a little more insight. I didn't know that much about pharmacy and what pharmacists can do before shadowing. But another thing to keep in mind is that once you make a decision, or once you think you have a decision made, you don't have to stick with that decision. You can keep your options open. So through college, like I said, I chose Vassar because when I went to the campus, I just enjoyed the atmosphere. I loved the people that were there. Even though they didn't have a pharmacy program, they had chemistry, they had other science classes that I knew that I could take and major in. And there were other things that led to my decision to go to Vassar, not just solely about what programs or classes they offered. So I liked that it was a smaller school. I liked that I could have a better relationship with professors there. So Vassar is a pretty small school. Instead of having classes that could have 300 people in a big lecture hall, a lot of my chemistry classes were 15 to 20 people. So I liked having a close-knit community. And so also through Vassar, I was able to take a bunch of different classes outside of science. So I took philosophy classes, I took religion classes, I took math classes. So through all of that, I learned a lot, but it helped me kind of confirm that science was the way that I wanted to go. So yeah, I think my advice would be try to learn as much as you can about different disciplines or jobs or majors, but also don't be afraid to explore outside of your comfort zone at first before you really figure out what you want to do.
That's great. I love that guidance. That's excellent guidance. And, it must have been also a period of time where you were exploring, you were at the same time as the other students who were exploring and learning not only about what you liked, but what was available too, and the discovery process of, what are career options or science research options. I mean, I think there's so much to be said about that discovery process too. You
Yeah.
mentioned something though that is very much in light with why we created the Purdue Lecture Hall series, was to expose high school students to what a lecture hall at university with 300 seats really looks like, and what does that feel like when you come in to a lecture hall that is that big. So Molly, we thank you for doing this on video, but you know that this would have been at a lecture hall with so many seats. Tell us a little bit about going from Vassar, where it might have been smaller, but then arriving at Purdue, where we do have some of these larger classrooms or lecture halls with 300 plus seats for students. What
Yeah.
was that like?
Yeah, it was definitely a culture shift. And going to a big university isn't necessarily a bad thing. I'm so thankful to be at a big university now. For example, for like my research purposes, where at Vassar, there would be only one instrument called an NMR. So an NMR is an instrument that we use to characterize or identify the molecules that we have made in the lab. So at Vassar, a small school, we had one of those instruments on the entire campus. Whereas here at Purdue, we have 6, 7, I don't even know. We have way more of this instrument that is available, and different types of this instrument, different sizes, they can do different things. Similarly, the chemistry department at Vassar was really small. There were just a couple professors, whereas here, not only in the MCMP department, but in the chemistry department, in the biology department, there are so many more professors who are doing so many different things with their research, doing different fields within chemistry, organic chemistry, physical chemistry, inorganic chemistry, just things that I really was never exposed to at Vassar just because it was smaller. So that definitely is a nice thing about Purdue, is that I've been exposed to a wider breadth of people and research interests and resources available for me. So that's been really nice that I've been able to have that here.
Totally. I agree 100% with you. It's just like a playground with so many different options, so many different ways of tackling your scientific questions, right?
Mhm.
Molly, you did such a great job at explaining not only your science, but some of these tools that you're using that are just really complex. And, I applaud you at taking the complexity and bringing it down to a level that we can understand, us mere mortals here that don't do drug discovery like you do. And I just, I want to ask you a little bit about what are some of the uh, classes that, some of the high school students should be thinking about if they wanted to go down this route where you're going. And this is, I mean, I just want people to understand that, Molly, what you do combines chemistry, it combines biology, it combines, drug discovery and this term medicinal chemistry, which is different than just chemistry. I mean, there is so much that goes in doing what you do. There's so many techniques. Biotechnology is involved, and cell cultures and all of this stuff. So, what are some of the classes that you say, oh, I wish I would have taken a little bit more of these to be better prepared for what I didn't know I needed to be prepared for?
Yeah, I think if I could go back, I would focus a little more on the biology side of things, because like you said, just because what I do in the lab is mostly medicinal chemistry, so it is pretty heavy in the chemistry. So majoring in chemistry, I pretty familiar with all of the things that I'm doing on that side of the lab. But yeah, like you mentioned, it does have a lot to do with the biology, especially understanding, cells and how, microbiology, what's going on in your body, how enzymes are able to metabolize things, what is actually happening inside a human cell. Right. So thankfully, I don't work with human cells as much because I'm working mostly with bacterial cells. But there are people in the department that I work in who their targets are within humans. So they have to be a lot more familiar with the physiological processes going on inside humans. So that definitely helps them being able to know both the chemistry side of things and the biology side of things. I also, once I arrived here, I took a biophysical course. So a little bit of physics involved, a little bit of math to understand some of the more technical workings of how, molecules bind to the proteins. But something that I hope is encouraging is that a lot of the stuff that I talked about here today was knowledge and understanding that I acquired during my studies and during my research once I got to grad school. And people who are in my department now come from all sorts of different backgrounds. There are people who majored in chemistry, majored in biology, majored in pharmaceutical sciences. But most of the things that are most important for me now, understanding what an MIC means and what a KI means, and how do I know that these molecules are effective? That's things that I have learned both in a classroom and then also like technical skills of, how do I grow bacteria? I never touched bacteria in my undergrad research. So as even though it is important to, try to take as many different classes as you can to gather all of this knowledge before grad school, at the same time, once you get to grad school, your learning curve, how much you learn and understand, will grow exponentially. And you don't have to have a specific background necessarily before going to grad school.
That takes courage to do what you're saying, meaning that, you're going to get to a point where you're going to figure it out, even if if you've never taken the course to grow bacteria. You're going to read about it. You're going to read the recipe. You're going to watch a video. You're going to really just pick it up and try it, and you're going to fail a few times. It's not going to work, and you're going to throw it in the garbage, and you're going to try again, right? Tell us a little bit about,
Hey, Dr. Soares.
Yes.
It's Abby. I'm still driving. I'm sorry.
No worries.
I just wanted to say thank you for pointing that out, because I oftentimes will talk to my students. My famous quote is, learning is hard, but they oftentimes feel overwhelmed by the amount that they don't know, instead of encouraged by the amount that they're learning. And so I thank you in particular for pointing that out, because a lot of them feel like they should have all the answers right now, especially as they head into college and know what they should study and what they want to become. And oftentimes, as you and I probably know better than others, it just changes. That path always changes, and you're always learning more and then doing new things. So Molly, thank you as well for pointing that out and encouraging them that it's okay to fall flat. As long as you fall forward, you're going to be just fine. So
you always learn a lot when you fail at something. Probably you learn more when you fail at something than when somebody pats you in the back and said, oh, you did such a great job at that. You're like, oh, I did. Oh, okay, great. But I think, Abby, thank you so much again for chiming in, because I wanted to point out that I see in your character, Molly, that you have courage to just, go get into this. We're going to figure this out. We're going to go, and not having the microbiology background didn't stop you from heading into this direction where now you're going to need microbiology to test the effectiveness of the drugs that you're making. And, I want to know a little bit about how can you inspire some of that courage? What can we do to give this next generation of scientists the courage to say, as Abby said, we're going to fail, we're going to fall flat, we're going to fall forward, hopefully, and learn from these things. But what is it that has given you the courage? And what were some of the scariest things that you thought of as you were transitioning from high school to start in college? And what gave you courage then?
Yeah, I think something that I take, even now, that I kind of take a lot of comfort in, is knowing that everyone is in the same boat as me in some way, right? So I think there are very few high school students, 17, 18-year-olds, who, while they're getting ready to graduate, they know every single answer to what their future may hold, right? They know exactly what college they're going to go to, what major they're going to have, what job they're going to have afterwards, what the rest of their career is going to look like, and I've noticed that even in grad school, that any fear that I have, every other grad student along with me also has that same fear, right? So when I'm feeling frustrated because I just failed an experiment for the third time, the person down the hall in their lab, they might be doing a completely different research project. They just failed an experiment for the fifth time. And, when I am struggling to understand a new concept, the person that I'm learning from, they struggled learning it the year before. So I think you have to get comfortable with being uncomfortable, which is really hard to do. It's a lot easier said than done. But just understanding that, like you were mentioning, you learn and grow a lot more through the failures and the struggles, and it's just part of the process. It's just something that, you have to go through the lows to hit the highs. And when you finally get that experiment to work, it's like, okay, all those failures were worth it, because now I know how to do it and I'm getting great results and I can publish a paper on this. I think the successes and finding the answers has definitely made me look back and be like, okay, it was all worth it in the end.
Absolutely. But Molly, we do have to go back every once in a while and say, wow, look at what I've been able to learn so far. Look what I've been able to accomplish so far. That's excellent. And I think even for high schoolers to say, boy, I have gone through all these courses. I have learned all these skills. I just have to keep going. And I don't know what would be your guidance for somebody coming into their undergrad, where it's pretty overwhelming in terms of the different classes. You're responsible for everything, and as you said, is there comfort in knowing that you're just sharing the same experience as everybody else that's there with you, and making sure? I mean, do you go and tell people, hey, I'm pretty scared about this stuff? I don't know what's going on. It's better honesty, isn't it?
Definitely.
being open with people and being able to share some of that, because I'm sure everybody feels the anxieties that we all go through as we start. But what are some of the things that, as people are preparing to go to college and maybe start on their first year, if they're thinking about doing research, what are some of the things that you think would help them uncover where can they do research and how to get started?
Yeah. If research is something that you want to get involved with at your college, whether you go to a small school like I did or a big university, regardless, I think the easiest thing to do is just, search, Purdue chemistry program, and then go through the faculty and read, a little bit about their research, just their little bios on the webpage, and just reach out to a professor, see if they would talk to you about their research, if they have any available space in the lab for you to go in and learn about, what they're doing. Because, professors, they're excited about their research. They want to get students involved in their research as well. So it never hurts to ask someone if, you can just talk to them about what they do, or ask, if you can shadow a student for a day who works in their lab, things like that. It never hurts just to put yourself out there and ask for more information, or, find an older student who does have experience and ask them how they got into it, if they enjoy it. Yeah, I think that's the big thing, is just trying to reach out to faculty.
Fantastic. Thank you so much, Molly. Molly Youse from the Department of Medicinal Chemistry and Molecular Pharmacology in Dan Flaherty's laboratory. Thank you also for providing your email. And maybe if somebody does have a question or wants to ask you, how do I get started in MCMP or doing this type of work in drug discovery, hopefully they'll reach out. Thank you again, Molly. This is awesome. I congratulate you on your fantastic work and great progress so far. And thank you for teaching us all of these great pieces of science, laboratory techniques, and sharing us about your journey so far and your experience so far. Wishing you much, much success on all of your scientific endeavors, for sure.
Thank you.
Thank you, Molly. All right. I think I